So what do we mean when we say physics of living matter? We mean that we are interested in living systems like bacteria, plants, fungi, animals, and we want to describe those using the language of physics. So talking about particles, fields, forces, energies, torques, fluctuations, instabilities, and so on. And you can of course look at these systems at very different length scales. You can, for example, zoom into individual cells and start to think about how do the molecules assemble inside the cells, and utilize the techniques from molecular physics and thermodynamics to understand that. On the other hand, you can take a look at individual cells and describe their behavior using, for example, fluid mechanics or mechanics of solids. And you can go even higher length scales. You can look at multicellular systems or populations and describe those using statistical mechanics. In this group context, I'm leading a research group called Active Matter here in Aalto University, Department of Applied Physics. And we are a very interdisciplinary group tackling this kind of problems on living matter. Many of us have background in experimental physics, but there are also people with backgrounds in theoretical physics, microrobotics, chemistry, and microbiology. Here you see some examples on what we are working on. So living systems like microalgae, mammalian cells, bacteria, but then you see also other things like electrohydrodynamics, magnetic fluids, and so on. So the key research questions in my group are that we are interested in understanding how living matter, so living cells, assemble and self-organize into larger structures, and how we can control this organization. And this is an important question because how the material functions depends very much on how it is assembled. Second question that we are tackling is that can we create simplified systems, synthetic systems that display the core features of living matter? And very often when we carry out our research, we have some sort of particles, sometimes very complicated ones, in liquids, and they interact, and something complex comes out of these interactions. And on the side of living systems, for us, for example, a single bacterium is a particle. It's a complicated micron-scale particle, but it's still a particle. Microalgae, even larger and more complicated particle. Now it does also photosynthesis. Mammalian cells, even more complicated and larger, also now deformable particles. Then on the synthetic side of things, we can work with almost anything. We can work with tiny nanoparticles, solid colloidal particles, emulsion droplets, reverse micelles, and so on. And we can very accurately control the properties of these materials. And then really understanding what comes out of these systems, you need to understand the interactions between these particles. And of course, these interactions vary a lot, from very simple steric interactions to van der Waals forces, electric double layer forces, hydrodynamics, magnetic forces, and so on. But really, from this kind of a picture of having particles interacting with themselves in fluid, really complicated and beautiful phenomena can arise. So for example, here on the top left corner, you see bacteria at low and high concentrations, and I will return to this topic later during this talk. On the bottom left corner, you see mammalian cells. So these are now sort of sticky cells. They like to stick to each other and form this kind of cellular aggregates that consist of maybe hundreds or even thousands of cells. And these behave surprisingly as viscoelastic liquids. And what we have done in my group recently is that we have shown that these living viscous droplets, they can form living capillary bridges between solid surfaces, just like normal liquids would do. And it's fascinating how this kind of a really complicated matter, its behavior reduces to very simple physics. On the top right corner, you see an example from the synthetic side of things. There we have very tiny magnetic nanoparticles that are being driven by electric and magnetic fields into very complex dynamic patterns. And here the nanoparticle size is almost 1,000 times less than the size of those structures that you see. And on the bottom right corner, you see example from our electrohydrodynamics research that I will also talk more about today. So let me start with the bacteria. And the key message that I want to tell you here is that the bacteria can be considered as self-propelled rods. And recently in my group, we have found out ways to control these living systems with magnetic fields. So if you open any textbook on microbiology, you will find drawings like this, showing the inner contents and structure of bacteria. There is the cell wall, cell membrane, DNA, proteins, and so on, maybe some flagella. But it is also well established now by physicists already some time ago that if you watch these objects from outside and look at just how they move around, you can consider them as rigid rods that move in the fluid. And the way they move is by pushing fluid backwards using the flagella that they have. And then it necessarily follows that they're also pushing the fluid forward with their body. So this is known as a pusher type microswimmer. And here you see actual bacteria under the microscope. So really rod-like hard objects moving around. Sometimes you see them colliding with each other and interacting with the hydrodynamic fields that they create. But then something more interesting happens when you go to higher concentrations of these active particles. You can, for example, see this kind of a behavior. And this is known as active turbulence in the literature because it reminds us of the classic inertial turbulence. But this is best described as this kind of a chaotic fluid flows caused by the bacteria. And this is actually an important feature of these living systems because this is not only seen with bacteria but with other living systems and synthetic systems. And even despite of 20 years of research into this topic, the mechanisms are still rather poorly understood and moreover the biological implications of this phenomenon. And part of the reason is that we don't really have good tools to interact with this kind of a living systems that move really fast and make complicated structures. And my research group has been developing recently magnetic methods to control these systems. And magnetism has the advantage that the magnetic fields penetrate living matter very easily, so they don't interfere with the system. But that's also the problem with them. You can't generate a notable magnetic response in a living system as such. But the trick that we do is that we take these normal, essentially non-magnetic living objects and we put them into a liquid that is weakly magnetic. And then this magnetic liquid translates the magnetic forces and torques on these active particles. And the physics is shown here in very short. So on the left-hand side, you see time derivative of theta, which is the swimming direction of a bacterium. And it is affected by two terms. The first one is a magnetic torque term that wants to align the bacterium with the external magnetic field. And then the second term is a noise term that comes from thermal fluctuations around the bacteria that want to disturb this alignment. And here you see the experimental realization. So first bacteria moving randomly in all directions, but immediately when the magnetic field is applied, they start to swim along the field. And we, of course, apply this to controlling this active turbulence also, like shown here. So on the left-hand side, you see the normal active turbulence. On the right-hand side, you see the case where the magnetic field is on. And you should see that on the right-hand side, the dynamics are much more slower, and there starts to build up a nematic alignment of the bacteria along the horizontal direction. And what this really then allows us to do is that we can prepare the bacteria system in this kind of a quiescent state with magnetic field on, and then suddenly turn off the magnetic field like shown here, so that we can directly visualize this hydrodynamic instability that leads to this famous active turbulence. And we can start to test different physics theories that describe this process. And we have also generalized this to different kinds of bacteria, and also to different kinds of organisms. So for example, to microalgae that are completely different photosynthetic organisms. So this really is pointing to the direction that these magnetic interaction methods are going to be a general tool for interacting with all kinds of living microscopic systems. The second thing that I want to talk today is about electrohydrodynamics and how we can use electric fields to drive some elementary lifelike behavior to simple particles and fluids. So no more biology anymore, much more simpler physics, and all here is based on concepts of electrohydrodynamics and electrorotation. And these are phenomena that happen when you have a solid particle or liquid droplet in a surrounding fluid. And if the properties of the fluids and particles are suitable, then the application of an electric field induces charge formation on the surface of these particles or the droplets. And then the same electric field creates forces on that charge density. And that can lead to, for example, particle rotations or interfacial deformations. And in principle, the charging is very simple to explain using classical physics, Gauss's law and Ohm's law. The tricky thing is to combine and connect that to hydrodynamics. And without showing the details, I'll just show the end result. So these are microscopic glass spheres that move by rotating on a solid surface, and these are propelled by the electric field that I just mentioned. And you also see some obvious interparticle interactions here. And now if I show you again this video about the E. coli, so there's an obvious similarity here. There's motility, hydrodynamic interactions. The difference is that one of these systems is based on very complicated biology, the other one very straightforward physics. And in my group, we are also striving towards putting more and more externally controllable interactions to these systems, and we can essentially command these synthetic systems to, for example, form this kind of a change state that are not typically observed in the living systems. We are also taking this idea a bit further. What you see here on this video is essentially the same system from previous slide, but we have replaced the solid particles with liquid droplets. And because the liquid droplets are deformable, they do not anymore behave as simply as on the previous slide, but instead they form what could be called as liquid worm-like states that propel them on the surface. And if you look at very carefully on the background, you see that these liquid self-propelled worms are actually born out of a thin liquid film in the background, and they fuse together, forming larger and larger, more stronger artificial swimmers. And in the end, not really shown well on this video, but they go find the bulk liquid and merge with that and sort of disappear from the existence. So at some conceptual level, this is a very simple life cycle in a synthetic matter. And that brings me already to the conclusions. So firstly, I want to mention that there is this progress towards understanding living systems using the language of physics. This is a very hot topic around the world, with research groups doing both experiments and theory and numerical calculations. My group has been active related to bacteria, microalgae and mammalian cells. And by doing this kind of basic research, we want to find out something fundamental about these systems that hopefully will change our understanding about bacterial infections, maybe cancer metastasis, and maybe some implications on even sustainability and photosynthesis. And then we are aiming at creating these synthetic systems with lifelike behaviors. I gave you examples of simple motility and interactions and a really simple life cycle as an example. And it really remains to be seen whether something important comes out of these efforts, but at least we know that these synthetic systems are excellent model systems for the actual living systems. And with that, I would like to thank everyone who has participated in this research over the years. So my current group members, past group members, and our collaborators around the world. And of course, thank you for coming and thank you for your attention.